Well, good afternoon, everyone. So happy everybody could join us for the first of a webinar series put on by the membership committee of the state chapter of the Wildlife Society. Greetings, everybody. I'm Bob Holzman. I'm the current president of the state chapter and really pleased to welcome you here today. Just a couple of quick announcements before we get started with our two guest speakers. First of all, I want to thank the membership committee, Lacey Hill Castern and Jenna Malinowski and Brian Haringa for their work in organizing this. Also want to thank Dr. Ballmann and Mandy Kamps for agreeing to speak to us today, and look forward very much to their presentations. Uh, while I have your attention, a quick sort of commercial plug for the state chapter of the Wildlife Society. We're a membership-based organization. I know that we have a number of members that are signed on today for the meeting, and perhaps some of you are not familiar with the state chapter. We're a professional association that is comprised of wildlife scientists, managers, educators, and conservationists from federal agencies, the Wisconsin DNR, our universities, especially those with major wildlife programs like UW-Stevens Point, Go Pointers, Madison, Northland College, and UW Green Bay, and others. And so, and conservation organizations as well. So, we're a place that provides professional training opportunities and development like you're participating in today. We're also a source of community. And frankly, if this year has taught us anything, it's the importance of community and being around some like-minded people. And frankly, we're just cool as hell. So if you're not already a member, I would encourage you to consider joining, checking out our website. Just Google search the Wisconsin chapter of the Wildlife Society. We have a site hosted by Wild Apricot. Check us out. If you like what you saw here today, you might also consider joining us for our annual meeting, which takes place the last week in February. And we have a number of speakers and an exciting plenary session scheduled that is going to focus on diversity and inclusion in the wildlife profession. So I encourage you to check us out. Hope you enjoy what you see here today and other places. So with that, I'm going to, I think, introduce our first speaker, who is a chapter member and, in fact, a former board member and a Pointer alum at that, Mandy Kamps. Thank you for joining us today.
Well, thanks for having me. And I will start sharing my screen and get all set up to get us going today. So, what I'll be talking about a little bit is giving an overview of the information, at least some of the information that is known so far about SARS-CoV-2 in animals. And for the most part, I'll just be talking about captive wild animals, the farm-raised animals, and wild animals. I won't talk a whole lot about domestic animals, but we'll hit on that just a little bit. So, but before we get into any of the information I want to show on animal detections, I just want to talk a little bit about the virus in humans. So as we all can remember, World Health Organization first learned of COVID-19, which is the name for the disease caused by the coronavirus, SARS-CoV-2, in the very end of 2019. And that was following reports from China that there were people there that were experiencing or exhibiting pneumonia symptoms. So talking a little bit about SARS-CoV-2 versus COVID-19. So I want to mention just a couple of things here just to show either the difference and also some of the similarities between them. So when we're talking about the virus itself, that's when we refer to it as SARS-CoV-2. And I do have a breakdown of what that abbreviation means. But when we talk about the virus, when we refer to it in infections in people, that's when we refer to it as COVID-19. So I just want to make sure that you're aware a little bit about the difference, or when we say SARS-CoV-2 versus COVID-19. And diseases are named, they do have names, because that does help enable discussions on a whole variety of things, from prevention, transmissibility of it, treatments that might be out there. So that's why there is a name for the virus itself, and also a name for the disease that it causes. So a little bit about coronaviruses in general. They are a very large family of viruses, and they are found worldwide. Some of them are found in human populations. There are many others that are found in animal species populations. And when it comes to animals, they tend to be found in more of a species or group specific disease. So there are a number of them that already are existing in both human and animals. And in some rare cases, some of them are zoonotic. So some of them can be transmitted from those animal populations to humans. So with SARS-CoV-2, still the source of it is still unknown, but there are reports and there are information that do lead us to believe that it is linked to a live animal market from China. A wet market is what they're called, like the one that is pictured here. And these markets are not really how we would think of like a farmer's market for us. But in these markets, there are live animals that are sold. Some of them are processed. Some of them are slaughtered there on site. There's a lot of water in these markets. There's a lot of other liquids. And so that's where that name of a wet market comes to be. And it is thought that this SARS-CoV-2 virus did originate from a similar virus from bats in that part of the world. They want to say there's different bat species that we have here that are not in China. And since SARS-CoV-2 had been discovered and identified, it is considered an emerging disease by the World Organization for Animal Health. So getting into a little bit about these detections in animals worldwide. So the first detections were in March of last year, and they happened to be in Hong Kong. And this was both in a domestic dog and a cat. And since then, you can see by looking on this map, there's a variety of other countries that have detections in animal populations. So this is one of the only times I'll mention that it could be either in a domestic animal, a farm-raised animal, captive wild animal, and then one of most recently, we have a wild animal detection in the US. So you can see the different countries. I don't list all of them here, but you can see them all on the map, some of the different species that are affected. And then if you look at the map, the bigger the circle that you see, the more cases and more detections have been in that area. So it is a very worldwide disease, even when it comes to animal detection so far. So narrowing it down a little bit to the first detections in the United States, it was back in early April of last year. The first animal that had tested positive for it was a tiger at the Bronx Zoo. And within a few days after that, the additional tigers that it was housed with and 3 other cats at the zoo also tested positive. The reason for them to be tested was that they did exhibit some unusual symptoms. So some of the respiratory illness symptoms that might have some similarities in our own human populations. And those symptoms were observed in late March. So if we can all remember back to that point, we were really just getting started with detections of the disease at that time, right around that time frame. And it was believed that the big cats at the zoo were exposed to a person that could have very well been asymptomatic for COVID-19. And a note to make that I'll mention here and then in a couple slides is the Bronx Zoo is a very big zoo. They are AZA accredited, and what that means is that they're already held to very high standards when it comes to animal care. And so they already have a lot of precautions in place. And with the more information known about COVID-19 in people, extra precautions were put in place with the animal care workers interacting with these animals. And it's an important note just to keep in mind is that even with all these extra precautions in place, things like this can still happen inadvertently. So switching gears a little bit to talk about some of the farm-raised mink detections. So getting back to more of the worldwide scale. First off, there was the first detection in the Netherlands, and this was late April of last year. And again, it is believed that the mink on these farms developed disease from either the farm owners, farm workers, or those who had visited the farm, somebody who was on the farm that had COVID-19 symptoms. As they moved towards the end of 2020, in total, there were 69 mink farms that were tested positive in the Netherlands. And then by December of 2020, the decision was made is that all of the remaining mink that were alive on the farms, some of the farms had previously culled some animals earlier in the year, but the ones that were remaining, that the decision was made to let them all reach until the end of the year to be pelted, and that there were no more mink present on these farms. So, and a decision that was also made that went along with this was that legislation was proposed and approved to end mink farming in the Netherlands, and this disease likely spread up that decision. So actually, just as a few days ago, there should not be any active mink farms in the Netherlands. And then in Denmark, back in June of 2020, that's when Denmark had their first mink farm test positive. And again, here it was believed that the mink on the farm were exposed to the disease from a person working or on the farm that had COVID-19. This initial case here, the mink did not exhibit symptoms and there was no increased mortality, but that interaction with that positive individual prompted some testing to happen, and that's how that detection was made. Initially, Denmark decided to cull some of the positive mink farms, and then they switched over to some other active surveillance on all of the mink farms in the country with not as much culling happening. But by November, early November of last year, there went up to 207 farms that were testing positive. So this decision at that point was made to cull all of the remaining animals on the farms in Denmark. Another note that that they had realized with this virus that was circulating in portions of Denmark was that there were some mutations that were happening once the mink were positive for the virus. And there was concern about that mutation then spreading back to the human population, the people living in the area, and then ultimately more concern about the vaccines that we have available now, that the vaccines would not be as effective with these new mutations. So that there was not just a concern for the animal health here, but also human health and what that could mean for some future implications. And that information also was a big factor in determining to cull the remaining mink in Denmark. So switching away from a worldwide aspect to our farm-raised mink detections in the US, we have 4 states so far that have had positive detections. In Utah, the initial case was discovered because of an unusual high mortality rate in the mink on a farm. And this farm also had workers that were tested as COVID positive. The farm in Michigan, the mink on this farm exhibited illness and an increased rate of death. In Oregon, again, I feel like I'm repeating myself here, but this farm also had workers or individuals on the farm that tested COVID positive. And this is one where the farmer reported it and was really proactive in making this report to the state to have mink tested. And then unfortunately here in the state, in one of our counties, we have 2 farms that tested positive, and many mink so far have died or been culled because of that. And I do have a link here on the bottom for some guidance that is available to farm-raised mink keepers. So this is on a couple of the websites, I believe it's CDC and USDA. They have some recommendations out there for mink farms. And what I like about the recommendations is that this really highlights that this is a One Health approach. So in the guidance, they have recommendations for people involved on the farm, whether they're working there, whether it's deliveries to the farm, and then also definitely considerations for the mink themselves and other domestic animals or livestock or even wild animals for that matter that may be present on the premise. So it really does capture that this is a One Health approach for the guidance that they're providing for the remaining farms that are still active. So in addition to the farm-raised mink, there are a couple of other animal cases here in the US that I do want to highlight. At the end of October, Zoo Knoxville had 3 tigers that tested positive. Again, they had very similar symptoms to the tigers from the Bronx Zoo, some of those respiratory symptoms. And here again, it is believed that the cats had caught the disease from an asymptomatic zoo employee. And very similar situation with the snow leopards at the Louisville Zoo back in December. So a few points that I know you're probably already thinking about is that we can see some clinical signs or symptoms in especially cats that do seem to be susceptible to the disease. And it really is believed that they are exposed to this from asymptomatic zoo employees. All of these zoos are AZA accredited, like I mentioned earlier with the Bronx Zoo. They are already held at high standards and they have implemented extra precautions, and the transmission of disease can still happen. So keeping this thought in mind at these very controlled facilities and captive settings, that something like this can still happen. So it should be something that we keep in mind as we either ourselves, as we work in this field, or others that may interact with any free-ranging wild animal populations, that this can happen. And most recently, there are a few gorillas at San Diego Zoo Safari that have also tested positive. And again, it is believed that they did catch the disease from an asymptomatic person, and they did exhibit some of those initial respiratory symptoms, which is likely the reason that they were tested to begin with. So this map was dated at the end of 2020. This is just the US, and it shows the different states and the number of animal cases that were detected by that date. So the number that you see on the map is just the animal case itself. It doesn't mean that that's the number of animals that were affected, because we know that with those mink farms, that involves thousands of animals. So our 5 detections that we have in Wisconsin includes our mink farms, and then also includes domestic cats and dogs. So lastly, we do have 1 case of a wild animal that has tested positive. And so far, this is the only known case of a free-ranging wild animal. So why this animal was tested was that as part of a One Health team, which again would involve agencies and organizations like USDA Wildlife Services, National Wildlife Health Center, state and federal agencies, both involved in wildlife, livestock or animal health, and also public health, is that teams are organized, and there was some surveillance or testing done in wild animals and other animals on those mink farms and surrounding those mink farms. So all happening in Utah, Michigan, Oregon, and Wisconsin is that the surveillance was happening where additional animal testing was occurring, because we wanted more information to know if other animals, what other animals, are they susceptible, just trying to get more information about the virus around the farms that have tested positive so far. And at that point, of the over 150 wild and non-native free-ranging animals that were tested around these farms, there was just that 1 wild mink that had come back positive. And it is not known yet, I know there's still a lot of work being done, but it is not known yet how the virus was transmitted from the mink farms, the animals on the mink farms, to this wild mink. So after going through just some of the cases, definitely not all of them, but I did try to pick out probably the more, the ones that we may be hearing about more, or as we work in our positions, whether it's with free-ranging wildlife or captive wildlife, or maybe even with the agriculture side, picked on some of those cases just to highlight and give some details on. But now I just want to touch a little bit about our general animal guidance when it comes to trying to figure out whether or not an animal should be tested. The differences here between animal testing and even thinking about the difference with when we do human testing. So there's not a lot of information known yet about SARS-CoV-2 in animal populations. However, from what we do know, it does appear that some animals are susceptible to the virus from an exposure with a person who has tested positive. It is less likely that the animals can then spread that virus back to a person or a variant of that virus, because SARS-CoV-2 can mutate. We know it mutates already, and to transfer that back to a person would likely have some mutations involved in that virus. We know of some clinical signs in animals, but we probably don't know what all of them look like. So some of them I do have listed in this table here, but you know, we're still trying to figure out and get information on which animals are susceptible to the virus, and even at that, which ones may exhibit clinical signs and what those clinical signs are. With veterinarians, when they care for cats and dogs, or maybe even other livestock animals, right now the guidance is that they are strongly encouraged to rule out other more common diseases before they pursue or inquire about getting that animal tested for SARS-CoV-2. So really trying to rule that out and saying that this routine testing for SARS-CoV-2 in any animal population is really not recommended at this time. If it is a consideration, it is taken as a One Health approach by some guidance that is out there to help professionals, not just in the domestic or livestock field, but also working with wildlife as well, to help determine if an animal should be tested. So this table might be a little bit small for everybody, but it is on the CDC website. And I just wanted to show that there are some criteria in this guidance that has been developed to show in certain situations, like if an animal has a history of exposure to a person or another animal that was suspected or confirmed to be positive for the virus, or maybe it's a threatened, endangered, or otherwise rare animal, or whatever that situation might be, working through those situations and then seeing as well, are there any clinical features that go along with that situation? What is that animal exhibiting? And given all of that information combined, is it warranted to get that, to test that animal for the virus? And this is, again, I know I've said it a few times already, but it really is a One Health approach with making this decision. So thinking a little bit more about wildlife guidance and testing, again, here, routine testing for this virus and any free-ranging or even a captive animal for that matter is still not currently recommended, may be considered in certain situations, and going back to those guidelines that I just talked about a little bit will help make those decisions. And here as well, just like with veterinarians treating domestic animals or livestock or other captive animals, before any wild animal that might be sick or found dead is tested for the virus, it is important to rule out any other common causes first. And lastly, thinking about the diagnostic test used for animals to do the testing to determine if they are positive, there's still not a lot of information out there about the sensitivity or specificity of these tests, or for that matter, how confident are we in detecting an animal, a positive animal, if it is positive, and how confident is it in detecting a, if you want to think about it as an animal that does not have the virus, how confident is that test in being accurate in coming up with those results? So when it comes to any animal or wildlife testing for the virus, there is still a lot of unknown information. even as we're still learning more about this virus in human populations, there's far less information known about it when it comes to animals. So what does this all really mean? Is that we know, based on some situations already, that there is potential for the virus to mutate in certain susceptible animals. This virus mutations could pose a risk to either other animals or even human health. And especially when we talk about human health, being cautious because those mutations may threaten how effective the vaccines are that we're finally being rolled out now. In general, disease is really difficult to control once it's in a wild population. So trying to prevent as best as we can a novel virus, an emerging disease as it's been classified, is even another reason to do what we can to put in protective measures so that this disease is not introduced into free-ranging wild animal populations. And ultimately, we just need to know more about this. And I know there's a lot of research projects going on right now, and we do have some information, and I think just continuing that work the best that we can. And lastly here, I just want to mention a few things that we are doing in the state here, some of our actions. So starting in spring of 2020, we have temporarily suspended the rehabilitation of bats, felids, and mustelids. We have also discussed any of the research or some of the field work that some of our staff or other agencies, others that work in the field here in the state, and how can we alter some of those protocols so that the handling or close interaction of certain animal or mammal species is limited. What kind of PPE can we add in? What are some other biosecurity measures? Do we temporarily hold off on a project or some field work for now until more information is known? So there are some of those modifications that we have been doing as well. There's many staff that have been involved in calls and meetings, not just with our other professionals in the state, but also with those working across the US, worldwide, state and federal, and a variety of different teams and groups that a lot of our staff are involved in just to stay up on the information that we know, and external communications. I know that we did work a lot on getting some communications out to the public about the rehabilitation suspensions. We also sent communications to humane societies, wildlife control officers, nuisance control officers, and those groups as well, as we know that they have interactions with certain animals that may be susceptible and that they could be taking extra precautions. So just wanted to list some of the things that we have been doing so far in the state. And with that, I am probably at my halfway mark. So I want to make sure I leave some time for Anne. And at the end, I believe is when we'll have some time for questions.
Okay, thanks, Mandy. We will questions at the end, and apologize here if I'm getting a little feedback on my microphone. I'm not sure what that's about, but anyway, Anne, if you're getting loaded and ready to go, we'll transition to you then. So I'd like to welcome Dr. Ballmann to the program today, and the floor is yours.
Great. Can everybody hear me? Because I can't see the screen. So can people hear me?
Yes, we can hear you.
Oh, super. Okay. And you're seeing my presentation. Is that correct?
Yes, we are.
Super. All right. So I want to thank the Wisconsin chapter of TWS for inviting me to speak today. There is quite a number of emerging diseases that have come up in recent decades, and time does not allow to obviously cover all of those. So what I'm hoping to do is at least touch base on highlighting some of the more recent emerging diseases where the National Wildlife Health Center has been conducting research on, and I wanted to expand a little bit on the presentation that Mandy gave as far as the COVID-19 outbreak and SARS-CoV-2 research that's going on at the National Wildlife Health Center. And in fact, I'm actually working from home because I'm in quarantine for a potential exposure from work itself. So again, it's circulating very widely. So good information to have on hand. So we'll be talking about SARS-CoV-2 as well as rabbit hemorrhagic disease, high path avian influenza, and white nose syndrome in bats. So what Mandy had brought up is back in the spring of 2000, when there was the pandemic, there was little information about the potential source of the virus, but it was being widespread, and more animals were becoming positive, or reports more animals becoming positive. Obviously, in the springtime, that's one of the peak times when white nose surveillance is occurring, and so a lot of managers and agencies had red flags raised with the pandemic raging, and potentially or planned bat handling activities occurring at that same time. And so one of the things that happened early on was that the National Wildlife Health Center, as well as with collaboration with Fish and Wildlife Service and EcoHealth Alliance, there was a rapid risk assessment that was performed, again, searching through the literature of what was best known about species that might be susceptible based upon what was known about the virus at the time, and comparing that or coordinating that with what was known about bat handling activities, is looking at 3 different groups of main concern. Obviously, the researchers and bat biologists that were going out in the field potentially handling these bats for monitoring purposes. Also, bat rehabilitation community, where there would be prolonged close direct contact and potential spread. And that was one of the main concerns, was the potential for asymptomatic humans to actually spread the virus into the North American bat population, setting up a potential reservoir that would be a constant source of reinfection in the future, not to mention not knowing exactly what the health effects would be on the bats themselves, which are already obviously challenged by, again, white nose syndrome and other threats to conservation. So in addition to the researchers, the bat rehab community, also wildlife animal control officers that may be interacting with bats. So what was done is they were looking at the best available information and solicited expert opinions for what the risk they thought would be for people who might be shedding, and determining based upon the amount of contact that they might have with a bat, what they judged would be the amount of risk. And overall, while there was a lot of uncertainty, the experts' conclusion was that there would be a non-zero risk of transmission potential. So there was some potential risk. It was hard to quantify with the available information, but it was enough to obviously want to take additional precautions and to consider what sorts of activities might need to be held off, or if there was to be activities occurring, ensuring that the experts did feel that the proper use of PPE, because not in all situations where PPE necessarily being used, would reduce the risk of spread. And so that was one of the things that the health center was involved with. Concurrently, realizing that there wasn't a lot of information available of what the susceptibility was in our bat populations to the SARS-CoV-2 virus. There were some infection trials, or there was an infection trial that was instituted at the health center back over the summer. This involved big brown bats that were actually collected in Wisconsin and infected with the SARS-CoV-2 isolate from the first SARS-CoV-2 patient in Washington State. Using that isolate, that what they looked at is they housed the bats in pairs. There were 16 bats in total that were tested. 7 of the pairs were housed together. One of the pair was inoculated with the virus both orally and nasally, and the second bat in the cage was not, to see if there was any transmission between bats as well. And then also compared to a sham inoculated pair. And basically looking at, over the course of 3 weeks, looking at all the samples, collecting, oral swabs, rectal swabs, tissues at set time points each week during that from the pairs to see if there was any evidence of infection. There was no evidence, at least in big brown bats from those infection trials, any evidence of infection or tissue virus invasion into any of the tissues, no clinical signs of disease, no pathology, and no evidence of transmission. That being said, though, this is, big brown bats, which are, at a higher risk of being in contact with folks because of they hibernate in human structures. They tend to be the most common bat species submitted for rabies testing. So there's that potential for close interaction with people. There are, 45 bat species in North America, and we know from other species that have wildlife species or animal species have been infected with SARS-CoV-2 that there can be variation among the different species within an animal group. So while we know it does not appear to be an issue in big brown bats, we can't say the same holds true for all. And so there is ongoing research right now looking at tissue cultures from other bat species, including little brown bats and Mexican free-tailed bats, looking to see if the virus will replicate in those tissues as well. So more information about that big brown bat infection trial was just published in Transboundary and Emerging Diseases in December 2020. And as Mandy mentioned, there are guidelines for testing in wildlife. One thing I guess I do want to highlight that at least for the bat submissions that are being submitted to the National Wildlife Health Center at this point, we do a coronavirus screening on those. Past pilot studies has shown about 10% prevalence of alpha coronaviruses in North American bat populations from bats that have been submitted to the health center. SARS-CoV-2 is actually a beta coronavirus, which is different from the alphas. That's where MERS and SARS and again COVID-19 comes from. The screening that we do for the bat submissions at the health center, we have to date not detected any signs of the SARS-CoV-2 virus in wild bats at this point. Some additional SARS-CoV-2 research, again, that was undertaken over the summer, again, out of immense concern when the virus was found to be responsible for mink mortality in Europe, was the concern by the Black-footed Ferret Conservation Recovery that their captive population would be at risk of potential exposure to someone who might be asymptomatic for the virus. So through emergency use, they submitted the request to attempt experimental vaccines. And so this was done in conjunction with Tony Roke's lab, who's also been instrumental in developing the Black-footed Ferret plague vaccine for prairie dogs and the recovery program. And so what their lab did is they developed an experimental SARS vaccine, and they went ahead and, because again, there was abundant concern about the potential for spread to the ferrets. They did vaccinate 120 of the black-footed ferrets at the center. They retained about 60 animals that did not have any vaccines because again, it was pretty fast turnaround time. And so they didn't want to, if there was any ill effects of the vaccinations, they didn't want to completely wipe out their population. So they did have 60 non-vaccinate controls. The animals that did receive the vaccine, it was an inactivated version of the virus. It was 2 doses were given sub-Q at 3 weeks apart, and they were able to demonstrate that the ferrets did exhibit antibody production as a result of the vaccination. They are not actively conducting infection trials in the population like we did with the big brown bats. So there's really no way to know the efficacy of the vaccine against the virus, nor the duration of protection. But at this point, they do know that the vaccine that was developed does appear to be at least safe to administer to the black-footed ferrets. And as far as to this point, there has been no detection of SARS-CoV-2 or mortality associated to the SARS-CoV-2 in the black-footed ferret population. So switching to another, leaving SARS-CoV-2 and switching over to another virus emerging disease that hit the landscape here in North America in 2020, rabbit hemorrhagic disease. This is a federal or foreign animal disease that was first detected in France in 2010. Sorry. Are you guys seeing my slides go through?
Yes.
Oh, okay, good. This was making sure. So it was first detected in France in 2010 and rapidly spread across Europe and replaced a related but certainly different virus, which is classic rabbit hemorrhagic disease. It arrived on North America in 2016 in Quebec. These were in that case, it was in domestic rabbits, farmed rabbits. When it showed up in France, it was both domestic and in wild rabbits. And like I said, it quickly replaced the rHDV2.1 variant that was out there. Most recently, and it popped up several more times in North America since 2016, but it was always in domestic rabbits or farmed rabbits. However, in March of 2020, there was an outbreak that first was detected or reported in New Mexico in domestic rabbits, but there was also concurrent ongoing mortality in the wild rabbit population that was later determined to be the same virus, which was rHDV2. And once it showed up in New Mexico, it's now to date, it's in 7 additional states. You see the list there on the slide, and those are in the order of which the cases were first reported for each of those states. In both cases, it has been reported in wild rabbit populations in all those states, the exception there being Florida, which would be a fairly far jump from the Southwest cluster from earlier this year, and it remains ongoing. The Florida case right now has been only in domestic rabbits that appeared in December. So obviously they are on high alert to look for the potential crossover into the wild populations as well. So the rabbit hemorrhagic disease is a highly contagious virus to both domestic and wild rabbits and hares. And in fact, all lagomorphs, including pica, are suspected to be susceptible. One of the differences between rHDV2 from the rHDV1 or classic rabbit hemorrhagic disease is that all age classes seem to be susceptible to rHDV2, whereas rHDV1 has really only ever been detected in European species of rabbits, and it tends to only affect adults and not the young rabbits as well. In North America, with this current outbreak, there are several species of jackrabbits that have been confirmed with rHDV2, as well as several species of cottontails, as well as feral and domestic rabbits. Feral rabbits being domestic rabbits that have escaped into the wild. So rHDV2, the incubation period is anywhere between 3 to 9 days after infection. Rabbits will start developing clinical signs and die. The die-offs can range anywhere between 1 to hundreds of rabbits reported, depending on the density in the area. The signs that are typically seen are lethargy, rabbits that are sitting out in the open and can be approached and not moving, labored breathing. You may see seizures, although most commonly, I think, reports are just sudden death, with the carcasses appearing to be untouched and just laying out on the landscape. And the photo here shows commonly these rabbits are reported with blood from around the nose and the mouth can be seen. Transmission of the virus itself is through direct contact with live or dead carcasses, but because the virus is present in all secretions and excretions, and the virus itself is very hardy, being able to survive freezing and can remain infectious up to 90 days. The meat and fur can also contain infectious agents. And so, again, this is highly contagious and can be spread really easily. So I have to be concerned about mechanical vectors, such as, predators on the landscape that may be moving the virus around, insect or other scavengers that can do the same, as well as contamination of clothing and shoes and food and water at a site that might be have infectious agents in it as well. So the RHDV2 virus is an RNA single-stranded virus, and as I mentioned, there are at least 3 variants known. The first to the RHDV-A or RHDV1, those are very closely related and have not ever been detected in wild rabbit populations here in the United States. Hence, foreign animal disease. And RHDV2 is what is currently circulating right now, mostly worldwide, because it has pretty much taken over and removed the RHDV1 from circulating. The virus that is involved in the die-offs right now that appear to be very closely related in the Southwest cluster, so Colorado, Arizona, New Mexico, Texas, and Utah. Those viruses are genetically very similar. There is some variation in some of the wild, the isolates that have been detected from the die-offs in California. So there is some mutation that appears to be going on at this point, and not sure of the exact source of that. The detection of the virus is through RT-PCR, most commonly in the liver, and all animals all the rabbits that are screened are also screened for RHDV1 as well too, when testing, just to make sure that that's not showing up on our doorstep as well too, and those have all been negative. Unfortunately, there are some vaccines available for RHDV2, but they are in Europe. We don't have any licensed vaccines in the U.S., and so those are being allowed for emergency use in situations where endangered rabbit populations may be at risk in areas where the virus is circulating in the domestic and wild populations. Because it was a foreign animal disease, all the detections, confirmatory testing originally had to be done through the USDA Foreign Animal Disease Laboratory in Ames, Iowa. And that still remains the case when the virus is identified in any new state or species. But since it's been circulating here now for several months and the number of cases has increased and it's becoming stable in certain areas, they've released the testing authorization for various laboratories, with the National Wildlife Health Center being one of those, being allowed to test the wild rabbit populations and do surveillance for the RHDV2. So we are one of the labs that are performing those tests for partners. And we'll accept rabbit submissions from new counties or species of unknown susceptibility in the areas where the virus has already been detected. As I mentioned, the virus is super contagious and highly resistant, and so there's lots of considerations to think about. Should it show up here in Wisconsin, ways not to have it transmitted unintentionally by our actions. And so, making sure that stringent biosecurity measures are in place, suitable PPE, boots, gloves, and coveralls, making sure that ideally any carcasses, rabbit carcasses that are found that appear suspicious with the clinical signs I earlier described, or blood around the nose and the muzzle, should be double bagged for transport to a diagnostic laboratory or for disposal that ideally would involve incineration or deburial because of the ability for the virus to survive so long. If you're handling any of these and you have pet rabbits at home, obviously you want to take precautions to shower and change clothes as well. Because rabbits are a common prey source or an important prey source in many ecosystems, there is concern about secondary effects of widespread rabbit mortality in the environment on their predator populations as well too. And they have seen some instances in Europe where when RHDV2 outbreaks have occurred that has had population level effects on some of these higher, some of the predator populations as well too. So some of the tools that the Health Center has been involved with, which RHDV2 has included doing risk assessments, looking at, density of domestic rabbit farms, that's shown there on the map in the lower right, as well as wild lagomorph richness on the landscape to see areas where would be at high risk for, potential crossover or spread into the wild populations, and just simply reviewing what techniques are out there for conducting rabbit surveys and making those available to folks in the field who might want to be doing some more occupancy data collection to help build those risk models. So I also wanted to switch gears or talk about another virus that has increased activity on the landscape, particularly here in this last month on this high path avian influenza, to be on the lookout for folks. Again, this is another virus. We are seeing increased activity now that was similar to what we saw prior to the detection outbreak back in 2014 when it entered the North America for the first time and resulted in the mortality in domestic poultry as well as wild bird populations. Currently, it's the high path avian influenza in 2020. It's been detected in 37 countries. The U.S. did have one detection back in April in South Carolina, and that was on a turkey farm with the subtype H7N3. However, there are at least 9 circulating subtypes of the virus that were reported in 2020, with the red ones being on that slide, the ones that have been detected in wild birds. And the species that are most commonly involved with that are, or the types of birds are swans, ducks, geese, grebes, shorebirds, raptors, owls have been reported so far. And again, because of the increased activity, we did release a wildlife health bulletin back in December just to have folks be on the lookout. And that was, here's some map that's showing the increased activity just in December alone. There was nearly 300 new outbreaks reported in wild birds across 17 countries in Europe, as well as some in Asia. About 90% of those were actually coming out of Europe, those new detections, as well as another 460 ongoing outbreaks that were still occurring. And as everyone probably is well aware, with fall migration and flyway overlap, there is the potential for the virus to enter the North American continent again. And so the alert was sent out to increase monitoring for any avian mortality involving waterfowl or raptors and other avian scavengers that might feed on birds that are infected and show signs, because that's one of the things we saw in the 2014-2015 outbreak, is that we were getting scattered reports of eagles and hawks that were, crash landing and subsequently dying, and were found to have be infected with high path avian influenza as well. So, should folks come across, avian mortality like that, large scale, or, certainly anything, 5 or more is what we usually tell people. They should be making sure that reports reports are going to at least, Wisconsin DNR Wildlife Health Program, if they're on state lands, or if federal or tribal, those reports can come directly to the National Wildlife Health Center. And again, importance to wear appropriate PPE and practice good biosecurity, because although human infection risk is potentially low, it is still possible. And with the bird submissions to the National Wildlife Health Center, we're continuing to do opportunistic surveillance and testing all the avian submissions, looking for avian influenzas, both high path and low path. And one of the interesting points of research that I wanted to point out associated with high path avian influenza is using artificial intelligence to help forecast potential areas of disease emergence or potential sites for high-path avian influenza detection, because flyways are very big and the migratory period is fairly long. So it's impossible to monitor everything. So trying to identify areas that would be places to focus on. They're using Doppler weather radar that can detect large flock migrations and link that to where poultry farms are as well too. So you can kind of see real-time movement of where the birds are moving and predict where hotspots might be and where to focus some of more enhanced surveillance activities. And this is a collaborative project between the Health Center, USDA, and the eBird at Cornell University. Real quick, since I see it's only 5 minutes left and it's not going to be much time for questions, I just wanted to point out some of the white nose surveillance in bats. Probably the most interesting thing for folks is with the COVID-19 outbreak, bat activities, direct bat handling activities were suspended. And so we were scrambling to still provide important information about PD prevalence, which is the fungus that causes white nose syndrome, where that might be moving into new areas on the landscape. And so using some data that was generated through pilot studies in Wisconsin in 2017, looking at using guano itself without direct handling of bats to screen for the fungus. We were able to apply the information that we learned from those studies that, again, were conducted in Wisconsin, where the white nose had already been present for several years, using that at the leading edge of PD, using that as a sampling mechanism for folks to screen or to survey for the potential range expansion of PD into those areas. We were able to do that and have that ability to pivot to that point this season, and were able to detect it in bats that weren't showing any clinical signs of white nose at the time, but did detect the Pseudogymnoascus destructans fungus at six roost sites in Montana, as well as 2 in Washington. I'm going to skip that, but just using the data from the about 10 years or more worth of surveillance data, modeling, looking at ways to more efficiently conduct surveys or where to allocate resources for white nose. We were able to do that with the modeling data and looking at landscape parameters that bats use, to where bats might be to generate where we want to target highest priority areas for early detection of the fungus itself. We're doing that at the National Wildlife Health Center as well too. And then for last day, I want to just bring up white nose vaccine research. We have in 2019, 2020, there were Wisconsin field trials done again by Tony Roke's lab, looking at testing 2 vaccine candidates against the white nose fungus in hibernacula. And they vaccinated about 300 bats at that point, and they had marked them so they could differentiate between the treatment groups for the 2 different vaccine trials tested, as well as the sham vaccinated bats, and looking to see how the vaccine would potentially improve or any differences in survival, as well as differences in pathogen load and shedding of the virus. And the vaccines that were applied appeared to be safe, with no differences seen among survival among the vaccine groups themselves. However, one of the vaccines tested did show that there had increased efficacy as far as reducing pathogen load and delaying the time of emergence from hibernation, suggesting that the physiological effects caused by a white nose infection were not as severe, so they stayed in torpor longer and were able to survive that. So the plans are going forward for next year is to repeat these studies, but actually apply the vaccine a little bit sooner during fall swarm to elicit stronger antibody responses in the bats. I know that we're at time, so I didn't know if we wanted to just stop here and open the floor for any questions that folks, or if you wanted me to just wrap it up in a single slide here.
I think you can wrap it up on the single slide, Ann.
Okay, all right. So just so folks know what is available, or when you would come across or how to get the most information about what's going on in your area. I just wanted to point out if folks weren't already aware of a WHISPERS database, which is an acronym for Wildlife Health Information Sharing Partnership. Event reporting system. This is a database that has information of the wildlife mortality events that have been reported or investigated by the National Wildlife Health Center, as well as other contributors are starting to put information into this database too from the wildlife management agencies. And it's a place to report sick and dead wildlife from state, federal, tribal natural resource management offices across the country. They provide online space for documenting and mapping these reports, so you can easily view by a map and see additional details by a table. These reports provide, the wildlife health community with a much better understanding of what is going on in the landscape, not only in your local area, but also beyond that, across the continent or even worldwide as well. So you can search it by species, by dates, location. There are different levels of access. Again, it's for wildlife managers, but public can access this at lower levels or, at county level information. If you have privileged access use, you can get more detailed information about location and the diagnoses, and folks are able to export data, create customized searches if they're interested in a particular species or a particular disease, and you can run those searches and generate reports, or again, spreadsheet tables that you can manipulate the data for your own purposes if you're tracking certain things on your end to do that. And for more information, you can go to the website listed up there. And if you're interested in more privileged access, you can request to be a registered user to actually be able to enter information as opposed to just view it, which is what the public has access to do. So with that, I will end my presentation and open it up for any questions.
Okay, so you will have to request to unmute. I did collect several questions in the chat box during the presentations. The first one was during your presentation, Ann, and it was whether or not there are any indications that rodents are vulnerable to SARS-CoV-2.
There has been a study that, and I haven't seen the actual published results yet, but I am aware of a study looking at deer mice, some infection trials, and I do believe, at least, and it's an open access report, so it's not been peer reviewed. They did indicate that they were able to demonstrate SARS-CoV-2 infection in deer mice. But again, it's not a peer-reviewed publication. It was just through open access, and I can find that citation and send that to this person if they want that.
Yeah, that was John Gilbert from Gliffwick that asked that question.
Okay.
And then Mandy, back when you were talking about COVID mutations and mink, do you know if there's been any research looking at COVID mutations in pet ferrets?
I can't, I don't know if there is. I'd have to look into some of the resources I have to see if there's any work being done in ferrets. I don't know. Ann, are you aware of anything?
Infection trials in ferrets. Yes, I believe there was some done in China. I don't know if there's anything been done locally, but I don't have the specifics on that information. I just know that they were able to demonstrate that ferrets are susceptible.
All right, and then the next question I have is from Shelley Dubé, and this is, Anne, when you were talking about our HDV2, is the Florida isolate different too?
They are still doing the genetic analysis on that one, so I do not know the answer to that. If it's similar to the genetic sequence of what was showing up in the Southwest cluster or California or something completely different, I do not know. I do know that the RHDV2 isolates that were detected in domestic rabbits that occurred earlier in the outbreak. There was reports in Ohio, in New York, and a third state, which is I'm forgetting at the moment right now, Washington, I believe. Those all were different sequences than what ended up showing up in New Mexico, both in domestic rabbits and then subsequently when it spread into the wild populations. So it's possible that the Florida one is completely different, or it could be similar. Don't know yet.
Okay, and then the next question is from Will Smith. Do zoonotic pandemics all have common patterns that are able to be combated?
Wow, that is a tough one. No. I mean, it's certainly going to depend on, whether it's viral, bacterial, fungal. There are lots of different emerging diseases that we didn't even touch on in those different groups of infectious agents, and some of the emerging diseases potentially are exacerbated by climate change as well too. And so it's I don't think there's going to be a one size fits all solution for trying to combat these global pandemics.
All right, thank you. And this is the last question that we've received in the chat box. Does Whispers database, this is from Andrew Edwards from Redcliffe, does Whispers database only contain records of confirmed or positive infections, diseases, et cetera, or does it also contain information about ongoing surveillance efforts that may be underway or planned? This sort of information would be helpful to researchers and managers as well as for the collaboration, project planning, et cetera.
Right. So currently, the Whispers database is set up right now for positive detections. We are working to on the surveillance component in what I would consider our phase 2 rollout of Whispers to incorporate just that, looking at looking at the surveillance data that would be available out there. So identifying where samples have been collected and tested but yield negative results. So that is in the works. It's down the road. I can't tell you exactly when that will be rolled out, but that is hopefully going to be added here in the next phase.
All right, well, that is all the questions we have right now. I really want to thank Anne and Mandy for being the presenters for our first web series for the Wisconsin chapter of the Wildlife Society. We will be planning on having these the second Wednesday of every month from noon to 1. More information will come, but the one that is being planned for February 10th will be on wild rice restoration and management. Like I said, more information will come on that, but please save the date in your calendars for the second Wednesday of the month from noon to 1. And thank you everybody for attending today.